I'm going to be talking about my research today, which is on the stream invertebrate assemblage composition in relation to environmental variables in the OESF. I'd also like to acknowledge that I'm working with Dr. Steve Bollins, Peter Kipney, Gretchen Wohlregen-Bollins, and also Kyle Martins. And also that this work is still in progress. I started this last fall. So macroinvertebrates are really important components of stream ecosystems because they form a critical link between primary producers of detritus and stream vertebrates, particularly salmonids. Macroinvertebrates are also really sensitive to any changes in the environmental conditions, and they're actually used as biological indicators of overall stream health just because they're so sensitive to the environment. And also because the macroinvertebrates in these headwater streams that I'm looking at tend to live in more isolated and heterogeneous environments. It's also thought that they're more heavily influenced by environmental filtering. And so down at the bottom here, you can see these are a few of the invertebrates I collected from the OESF streams. On the left are cranefly larvae. The top is Hexatoma, and then the bottom is Stapelidae. And then on the right, this is a stonefly larva. And so overall, my research questions were, what are the factors and processes structuring perennial headwater macroinvertebrate assemblages in the temperate rainforests of the OESF? And then also specifically, I'm trying to parse out the relative roles of dispersal and the environment in structuring these assemblages. And so I started this project in August of 2020, where I hiked out to the different stream monitoring sites that the DNR is monitoring for the T3 project that you'll hear about later today. And so there were 32 different sites that I hiked out to. One of them was dropped, so 31 sites, and that's over 16 different watersheds. So there was 2 sites per stream, one adjacent to where the riparian treatments will be for the T3 project, and one downstream at the pour point of the lowest point of the watershed. And so at each site, I collected a few different kinds of samples. The first one was of drift invertebrates, where I put a drift net right here in the main portion of the stream and let it sit there for 30 minutes to collect any invertebrates that were drifting through the stream. And then I did 1 or 2 of these depending on the width of the stream at each site. After that, I collected benthic invertebrates using a Haas sampler, as you can see right here. These are quantitative samples of the invertebrates that live in the bottom of the stream. And I did this by just disturbing the substrate and letting the disturbed invertebrates flow back into the net right here. And I collected 5 of these Hess samples per site. And then lastly, I collected microalgae samples, specifically periphyton and cestin. So to get the periphyton, I essentially just scrubbed cobbles and then let water run over them so I could collect the water and all the microalgae growing on the cobbles. And then I also collected a couple of GRAT samples from each site to get the cestin or the phytoplankton, the microalgae that's growing in the water column itself. And so right now I'm going through, I guess I should say, the results I have right now is that I collected in total 148 benthic samples, 39 drift samples, 103 periphyton samples, and 60 cestin samples. And so it turns out that there's a lot of lab work associated with these. And so right now I'm in the process of all of that. So I'm going through and doing what's called bug picking, where I'm pulling out the invertebrates from the rest of the, I guess you could say, debris in my samples and sorting it by order. And that way later I can go back and sort it to the lowest possible taxonomic unit. Typically this is genus or species, but for some more difficult to identify taxa, like oligochaetes or chironomids, it'll be suborder or family level. I also have already completed the lab analysis for the algae samples, where I filtered the algae and then used fluorometry to determine the chlorophyll A concentrations. And then lastly, I plan to use multivariate statistical analysis, specifically non-metric multidimensional scaling, to analyze these assemblages. And so hopefully I would like to look at, well, what are the relative roles of the environment and dispersal specifically just because this is not really well known yet, especially the role of dispersal for structuring macroinvertebrate communities in headwater streams and temperate forests. And then also I'd like to pinpoint which environmental factors are most closely associated with the macroinvertebrate assemblages. And hopefully this will allow the DNR and other natural resource managers to prioritize specific aspects of habitat quality for conservation. And then lastly, as I was alluding to earlier, this work is part of the pretreatment monitoring for the T3 project, which you'll hear about later. I was sampling at the same sites that they're doing fish studies for, and then later, after the treatments, the riparian harvest treatments are set in motion, we'll go back and sample the macroinvertebrates again.